Hello, and welcome to Mysteries of Earth. Today, we are stepping into a world so alien, so distant, that our modern senses can barely comprehend it. Billions of years ago, the Earth you know did not exist. There were no familiar landscapes, no forests, no mountains carved by time. The planet was still young, its surface battered, chaotic, and restless. Imagine a place where the oceans were not clear or inviting, but murky and metallic, tinted by dissolved minerals that glinted like hidden treasure beneath a dim sun. The air was thick and hazy, a heavy mix of gases that would have been poisonous to us, yet perfectly natural for the earliest forms of life. The sky itself was not a soothing blue. It might have shimmered in muted oranges, reds, or even greenish hues, a filter of methane and other chemicals suspended in the atmosphere. Sunlight was weaker, and yet it mattered more than ever for the life below. The continents were beginning to take shape, but they bore no resemblance to the familiar outlines on a modern map. They drifted slowly across the molten mantle, colliding, breaking apart, and reforming in a dance that would span hundreds of millions of years. The landmasses were low, barren, and jagged, volcanic crags rising from a world largely devoid of soil. No trees clung to their surfaces, no animals prowled, no birds painted the sky. Instead, a stillness dominated, punctuated only by the occasional volcanic eruption or meteor impact. And yet, life was present. It was small, single-celled, invisible to the naked eye. These microorganisms were already thriving, and in their quiet persistence, they were performing some of the most consequential experiments in Earth's history. They had been around for billions of years, but during this eon, something remarkable was beginning to happen. They were learning how to capture energy from the environment, how to cooperate, how to shape the very chemistry of the oceans. Picture the oceans themselves, vast dark basins filled with waters that lacked oxygen. Iron floated freely, dissolved and shimmering, waiting for the first hints of chemical change. These waters were alive in a subtle, invisible way, bustling with microbes that metabolized sulfur, nitrogen, and other compounds. They were small, slow, and persistent. Some floated, some clung to rocky surfaces, and some formed the earliest layered mats that would one day fossilize as the first hints of organized life on Earth. Beneath the water, gradients of light, chemistry, and temperature guided the behavior of these microscopic organisms. Some thrived near the surface, basking in the faint sunlight. Others retreated into deeper waters, into chemical shadows, where life had to adapt to extreme conditions. Every microbe had a role. Some released waste that became food for others. Some altered the environment in subtle ways that would ripple across ecosystems. Together, they formed the first ecosystems, complex, layered, and entirely alien. Above them, the early atmosphere was a theater of chemistry. Methane, carbon dioxide, nitrogen, and traces of hydrogen hung heavy in the air. Ultraviolet radiation from a young sun poured in unfiltered, making the surface dangerous for anything unprotected. Life stayed underwater, where the ocean acted as a shield, softening the sun's glare and stabilizing temperature extremes. The continents were largely inert players in this story. Bare rock, slowly eroded by wind and water, occasionally reshaped by volcanic activity. But Earth was changing. Slowly, over millions of years, subtle shifts began in the oceans. Some microorganisms discovered how to harness light in a new way, splitting water molecules and releasing oxygen as a byproduct. At first, this oxygen seemed inconsequential, disappearing into iron-rich waters and volcanic gases. Yet, Every bubble released, every tiny change, laid the groundwork for a transformation that would alter the planet forever. The continents themselves were being sculpted by processes we now call plate tectonics. Cratons, the ancient stable cores of continents, began forming, laying the foundation for the continents we recognize today. Some of these cratons persist under North America, Africa, and Australia. They are remnants of a time when Earth's surface was still learning how to hold itself together. Mountains were rare and low, rivers were short-lived and erratic, lakes formed in impact craters or along fractures in the crust. It was raw, elemental, and utterly alien. Even at this microscopic scale, life was already experimenting with cooperation. Cells merged, exchanged genetic material, and built structures too small for any eye to see. They were chemical innovators without intent, unaware that their metabolic experiments would one day make oxygen the most transformative force in Earth's history. Imagine standing on the barren land, looking out over these ancient seas. You would see nothing larger than a speck in the water, yet all around you, life was busy, shaping the future. Every chemical reaction, every microbial mat, every oxygen bubble drifting through the iron-laden ocean was a chapter in a story billions of years in the making. The Proterozoic was not a quiet or passive era. It was an age of slow revolutions. Oceans rusted, atmospheres shifted, microbial communities evolved in response to subtle chemical gradients. The stage was being set for the rise of complexity, for the creation of conditions that would allow multicellular life to flourish hundreds of millions of years later. Rocks from this time, banded iron formations, red beds, and ancient stromatolites, whisper hints of what was happening. They tell us that oxygen was seeping into the oceans, that continents were consolidating, and that life, though invisible, was already powerful enough to change the planet's chemistry. In this world, survival was about efficiency, about cooperation, about adapting to conditions far more extreme than anything we experience today. Yet, in this alien environment, life persisted. And in its persistence, it set the foundation for everything that would follow. So, if you are ready, let us drift further into this ancient realm. Imagine drifting along the shallow coastal waters where microbial mats clung to rocks, capturing sunlight and reshaping chemistry with every breath. Picture the slow transformation of oceans from iron-rich, oxygen-poor basins into landscapes that could one day host complex life. And above it all, the hazy sky, gradually shifting, hinting at the profound changes to come. The Proterozoic is not a time measured in years or centuries. It is measured in the slow, patient pulse of life altering its world. And as we follow this pulse, we will uncover how the first faint breaths of oxygen began a chain reaction that would eventually make Earth habitable in ways unimaginable at the time. Next, we will dive into those oceans themselves, the chemical landscapes, the first oxygen-producing organisms, and the invisible engineers of a world in transformation. Because to understand life on Earth today, we must first witness the world that existed before it even had a name. Oceans without oxygen. Imagine plunging into a world where the oceans are not the familiar blue expanses we know today. Instead, they are dark, heavy, and alien, rich with dissolved iron, sulfur, and other minerals that glint like faint fire beneath a muted sun. These were the oceans of the Proterozoic, a billion years before life would venture onto land, before complex organisms roamed the seas. The waters were anoxic, completely devoid of oxygen, And yet, far from lifeless, they were buzzing with invisible activity. Beneath the still surface, invisible revolutions were underway. These waters were thick with chemical gradients, a layered labyrinth of nutrients and energy, waiting to be tapped. The sunlight that penetrated the hazy atmosphere barely reached the surface, scattering in muted reds, greens, and golds. Imagine a shallow coastal area where the water appears rusty, almost metallic, reflecting the iron suspended in its depths. Deeper down, darkness enveloped everything, but life thrived in its shadowy corners. Here, microorganisms formed the earliest chemical networks, performing metabolic dances that would eventually reshape the planet itself. The dominant life forms were simple, single-celled microbes, yet their capabilities were astonishing. Some fed on sulfur, extracting energy from chemical reactions invisible to the eye. Others processed iron, capturing electrons from dissolved minerals and subtly altering the chemistry of the oceans. Still others consumed methane, releasing gases that drifted into the air above, adding invisible fingerprints to the evolving atmosphere. These early metabolisms were not just survival strategies. They were experiments in planetary engineering. Each chemical reaction was a tiny act of transformation, an invisible influence on the world around it. Visualize this alien seascape. Shallow lagoons stained deep red from iron presses, phytates, microbial mats spreading across rocky surfaces like carpets of green and brown. Tiny bubbles rise from the depths as gases escape from the metabolic work of invisible organisms. Layers of sediment settle slowly, creating banded textures in the rock beneath, signs that billions of years later, geologists would read as fingerprints of early life. The water itself is dynamic in chemical terms, though almost motionless to our eyes. Currents stir only slightly, but chemical gradients create a hidden flow, guiding the movement and behavior of microscopic communities. In these waters, life was both delicate and resilient. Microbes clustered in colonies, forming biofilms and mats that could withstand shifts in chemistry, temperature, and light. Some thrived near mineral-rich vents, their metabolisms tuned to the chemical bounty that seeped from the crust. Others floated in the open waters, harvesting faint sunlight or drifting on chemical gradients. Every layer of the ocean was a miniature ecosystem, invisible yet complex, a slow-motion ballet of survival and adaptation. The colors of these waters tell a story of chemistry and life. Rusty reds from iron, deep greens from microbial pigments, and browns from decaying organic matter paint a palette unlike anything we see today. The interplay of light and dissolved minerals creates shifting patterns, like a living painting in constant transformation. Even though the world above is barren and quiet, the oceans below are alive with chemical energy, shaping the destiny of life on Earth. Beneath the surface, oxygen was almost non-existent. This lack of oxygen shaped everything. Organisms had to rely on alternative strategies, using sulfur, iron, and methane as energy sources. Some microorganisms produced compounds toxic to others, while some formed symbiotic relationships, relying on neighbors to survive. These early interactions were the first whispers of cooperation and competition, the invisible drama that would eventually lead to more complex life. Even in these harsh alien waters, microbial ingenuity knew no bounds. Stromatolites, layered structures formed by microbial mats, began to emerge in shallow seas. They were not grand reefs, not yet. But these living carpets were engineers of their environment, trapping sediments, altering chemistry, and creating niches where other microbes could flourish. Their growth was slow, patient, measured in millennia, yet their impact would ripple through eons, leaving fossilized evidence for us to uncover billions of years later. The chemical revolution of these oceans was subtle but monumental. Oxygen, the molecule that would one day dominate the atmosphere, was being produced in trace amounts by early photosynthetic microbes. At first, it was absorbed into iron and sulfur compounds, invisible to the world above. But gradually, the oceans became saturated, and oxygen began to accumulate in localized pockets. Each bubble of oxygen was a tiny victory, a quiet revolution in a world dominated by anoxic darkness. It is tempting to imagine the surface of these oceans as silent and inert, but that would be deceiving. Waves lapped against barren shores, volcanic vents hissed and bubbled, and chemical reactions flickered invisibly beneath the water. Life was adapting, experimenting, reshaping the ocean from within. The Proterozoic oceans were laboratories of evolution, teaching organisms how to capture energy, how to cooperate, and how to alter their environment in ways no single species could achieve alone. These anoxic seas were also a mirror of Earth's own volatility. Iron-rich layers accumulated on the seafloor, recording chemical pulses from microbial activity. Shifts in temperature or chemistry could trigger blooms of particular microbes, which in turn would change the composition of the water. Life and environment were inseparable, engaged in a delicate feedback loop that it defined the trajectory of the planet. By studying these ancient oceans, scientists today read a story written in minerals and fossilized mats. They see evidence of microbial ingenuity, the first experiments with oxygen production, and the earliest chemical ecosystems. Each layer of rock, each iron formation, is a chapter in the hidden history of life, revealing that complexity begins not with visible creatures, but with invisible networks of energy and adaptation. As we drift through this alien world, it is humbling to consider how much of Earth's future was being written here, unseen. These invisible revolutions, occurring in dark, iron-rich waters, laid the foundations for the oxygenation of the planet and the eventual rise of multicellular life. The Proterozoic oceans were silent architects of destiny, patiently, persistently, transforming chemistry into opportunity. And as the first bubbles of oxygen began their slow accumulation, the stage was set for a transformation so profound it would redefine Earth itself. What began as subtle, invisible chemistry in murky waters would eventually ripple upward, altering the atmosphere, the continents, and the very potential for life. Next, we will explore that transformation, the rise of oxygen itself. How did a tiny metabolic innovation by microbes become a force capable of reshaping an entire planet? The oceans of the Proterozoic were the stage. The actors were invisible, and the drama was only beginning. The rise of cyanobacteria in the deep shadowed waters of the Proterozoic oceans. An extraordinary revolution was quietly unfolding. It was not a battle of giants, nor the clash of continents, but a subtle, microscopic phenomenon with consequences so vast it would ultimately transform the entire planet. The agents of this transformation were cyanobacteria, tiny, unassuming microbes that, in their own imperceptible way, held the power to reshape the Earth's atmosphere, its oceans, and the very possibilities of life itself. Cyanobacteria are remarkable in their simplicity and their ingenuity. These microscopic organisms harnessed sunlight through a process known as oxygenic photosynthesis. Unlike earlier microbes that relied on sulfur, iron, or methane for energy, cyanobacteria tapped directly into the sun. They captured photons, splitting water molecules into hydrogen and oxygen in the process. This might sound like a modest feat. After all, what can a single-celled organism do? But when multiplied across oceans, over millions of years, the result was a slow but unstoppable transformation of the planet's chemistry. Imagine billions of tiny cells drifting just below the surface, each a miniature solar panel, quietly converting light and water into energy and releasing oxygen as a byproduct. This was no sudden explosion. It was a patient, relentless accumulation, a whisper that would eventually become a roar. Oxygen began to infiltrate the oceans, initially reacting with dissolved iron, forming rust-colored layers that sank to the seafloor. These layers, known today as banded iron formations, are the geological fingerprints of this ancient, invisible revolution. Each stripe of iron tells a story of oxygen slowly, methodically reacting with the environment, marking the gradual awakening of a new planetary chemistry. The oceans themselves were changing. Once dominated by dark, anoxic waters, they began to exhibit chemical gradients shaped by the presence of oxygen. Microbial communities adapted to this new environment, some thriving where oxygen appeared, others retreating to deeper oxygen-poor layers. Cyanobacteria were the architects of this shift, not just by producing oxygen, but by altering the very rules of the game. In these hidden microbial laboratories, life was learning to harness energy from light and transform its surroundings, laying the foundations for future complexity. The colors of this world were shifting too, though in ways invisible to most eyes. Iron-rich waters still reflected deep reds and rusts, but tiny green mats of cyanobacteria glimmered near the surface, absorbing sunlight and releasing oxygen. Streams of bubbles rose from shallow pools, tiny exhalations of a world in transition. These were not grand rivers or crashing waterfalls, but delicate, persistent signals of life, altering the chemistry of the planet, one molecule at a time. This process was slow, measured not in days or years, but in eons. Oxygen did not flood the oceans overnight. At first, it was absorbed almost entirely by iron and other reducing compounds, leaving the waters deceptively calm, deceptively similar to the anoxic seas of the past. Yet beneath this quiet surface, a monumental shift was underway. Cyanobacteria were quietly transforming Earth's atmosphere, creating the conditions for an event that would be remembered billions of years later, the Great Oxidation Event, or GOE. The rise of cyanobacteria also reshaped ecosystems in ways that are hard to imagine. Before them, life was limited by the availability of chemical energy in the dark, iron-rich oceans. With the introduction of oxygen, new ecological niches emerged. Microbes could now exploit different metabolic pathways, adapt to new chemical landscapes, and even begin to interact in more complex ways. Oxygen, once a rare byproduct, became a new form of currency in the microscopic economy of life. The impact of these tiny organisms was nothing short of planetary. Oxygen began to accumulate, initially in the oceans, then gradually in the atmosphere. For the first time, the Earth had the potential for a breathable sky. Yet, this was no gentle process. Rising oxygen levels were toxic to many existing microbes, triggering extinctions and reshuffling ecosystems. The very act of cyanobacteria thriving created a paradox, life generating a force that challenged its own survival. This push and pull of creation and destruction is a theme that will echo through Earth's history, from microbial mats to the largest creatures that would ever walk the planet. Geologists studying the remnants of this era see evidence in the rust-colored bands of ancient rock, in the traces of microbial mats, fossilized in sediment, and in the subtle chemical signatures embedded in ancient minerals. These clues tell a story of patience, persistence, and planetary transformation. They remind us that even the tiniest forms of life can wield influence beyond imagination, shaping climates, oceans, and the trajectory of evolution itself. Visualizing this era is a challenge, for it defies our modern expectations. There are no towering coral reefs or schools of fish. Instead, there are subtle swirls of green mats on shallow shores, delicate layers of sediment accumulating on the seafloor, and a faint invisible cloud of oxygen beginning to pervade the water. It is a world that seems static and quiet. Yet beneath its surface, chemistry, light, and life are engaged in a ceaseless dance. Cyanobacteria remind us of the power of the microscopic. These organisms were not dramatic. They were not spectacular in the sense of visible grandeur. Yet their legacy is colossal. By capturing sunlight and splitting water molecules, they wrote the first chapter in Earth's oxygen story. Without them, the planet would remain a barren, anoxic world, with life confined to hidden chemical niches. Their quiet revolution would set the stage for more complex organisms, for ecosystems capable of supporting diversity unimaginable in the Proterozoic seas. And as oxygen slowly accumulated, it laid the groundwork for one of the most dramatic transformations in Earth's history. The oceans, once rust-colored and anoxic, began to host new chemical landscapes, while the atmosphere itself began a slow evolution toward oxygenation. Cyanobacteria had achieved something extraordinary. Through millions of years of patient persistence, they were engineering a planet. But this transformation was not yet complete. The oxygen they produced would eventually escape the oceans, saturating the atmosphere and triggering what geologists call the Great Oxidation Event. This was a time of both creation and destruction, of ecological upheaval and planetary change. What began as microscopic activity in shallow seas would ripple outward, altering the chemistry of oceans, the composition of the atmosphere, and the destiny of every organism to come. As we peer into these ancient waters, it is impossible not to marvel at the scale of what seems almost invisible. Cyanobacteria may have been tiny, but their impact was enormous, a reminder that even the smallest actors can drive monumental change. The rust-streaked oceans, the green mats glimmering near the surface, the bubbles of oxygen quietly rising. These are the early signs of a world preparing to breathe. And so, as the sun continues to shine over these alien seas, the stage is set for the next dramatic chapter, the Great Oxidation Event. How did Earth's atmosphere transform from a nearly oxygen-free sky into one capable of supporting diverse and complex life? What consequences did this sudden chemical upheaval have for the organisms already thriving in the seas? The answers lie just beyond the horizon, in the invisible forces shaping the fate of our planet, rusting seas and chemical revolutions. Imagine standing on the shores of an alien ocean, where the waters are not the familiar blue we know today, but deep, dark, and tinged with rust. Billions of years ago, the Proterozoic seas were a world transformed by unseen forces, a chemical theater where life and environment were locked in a subtle, invisible dance. The actors were microscopic, yet their impact would echo across time, leaving behind geological fingerprints that speak to us billions of years later. This is the story of Earth's rusting seas, a tale of chemical revolutions that set the stage for life's next act. The evidence is etched into rock, hidden in the crust beneath our feet. Geologists call it banded iron formations, or BIFs, layers of iron-rich minerals interleaved with silica, deposited in rhythmic patterns over millions of years. These formations are not just static rocks. They are the story of Earth's first oxygen. Each stripe represents a period when microscopic organisms released oxygen into the oceans, only to have it react almost immediately with the abundant dissolved iron. The result? Rust. Literally, rust settling in thick, orderly layers on the seafloor, a testament to chemical processes invisible to the naked eye. But why did this happen? The answer lies in the cyanobacteria, the tiny architects of oxygen. For eons, they had been producing oxygen through photosynthesis, splitting water molecules and releasing this potent gas as a byproduct. At first, the oxygen was absorbed by iron and other reducing compounds in the oceans, leaving the atmosphere largely unchanged. The seas were effectively a giant chemical sponge, soaking up oxygen and transforming it into mineral deposits. The red bands in BIFs are the remnants of this process, evidence of a world in transition, a planet quietly, irrevocably changing beneath its still surface. Picture the oceans during this time. The water is dark, slow-moving, dense with dissolved iron. Sunlight filters weakly through shallow layers, illuminating thin mats of cyanobacteria, performing their delicate alchemy. Tiny oxygen bubbles rise, only to vanish as they react with iron. Slowly, layer by layer, the ocean floor accumulates these chemical signatures, a record of the patient, relentless work of microscopic life. The seas are rusting, yes, but also transforming, becoming laboratories for experimentation on a planetary scale. This chemical revolution had profound consequences for life. For the organisms that had thrived in oxygen-poor waters, the increasing presence of oxygen was deadly. Oxygen is corrosive at the cellular level, toxic to many anaerobic microbes that had dominated the Proterozoic oceans for millions of years. Extinctions ripple through these ancient communities, removing dominant species and opening new ecological niches. Yet, at the same time, life finds a way. Microbes evolve strategies to survive. Some retreat to oxygen-free layers, some develop tolerance, and others exploit the new opportunities oxygen provides. The ocean becomes stratified, an upper layer enriched with oxygen, a lower anoxic layer still harboring ancient microbial life. The chemistry of the oceans is now a stage for life's next act. Oxygen is not just a toxic byproduct. It becomes a driver of complexity. It enables new metabolic pathways, allows for greater energy extraction from chemical reactions, and sets the stage for organisms capable of more ambitious forms of life. The ocean's transformation is slow, imperceptible in a human lifetime, yet profound in its implications. The rust-colored waters are a visual metaphor for change. What was once inhospitable is becoming a crucible for innovation and adaptation. As oxygen accumulates, the oceans themselves act as a regulator, moderating the pace of change. Iron-rich layers continue to precipitate, but gradually, the sponge begins to saturate. Oxygen begins to linger longer, spreading from shallow coastal regions to deeper basins. This sets off a chain reaction. More oxygen in the water, more selection pressure on life, more chemical diversity. The Earth is entering a new phase, where chemistry and biology are inseparably entwined, each shaping the other into subtle feedback loops. Visualize this world: the sun reflecting off shallow, rust-streaked waters, green mats of cyanobacteria glimmering near the surface, bubbles of oxygen rising lazily through the water, occasionally breaking free into the atmosphere. Deep below, layers of iron-rich sediment settle in ordered bands, preserving the chemical history of each moment. It is a world of contrasts, of stillness and transformation, of invisibly powerful forces shaping the very fabric of life. The BIFs are more than geological artifacts. They are messages from a time when life began to assert its influence on the planet. They tell us that the rise of oxygen was not abrupt, but gradual, a series of chemical revolutions played out over millions of years. These formations provide a window into the past, revealing the interplay between biology and geology, between life and the elements themselves. They remind us that even the smallest organisms can drive planetary scale change. Yet, the accumulation of oxygen also foreshadows dramatic consequences. As the oceans become increasingly oxygenated, the atmosphere too begins to change. Oxygen eventually escapes from the water into the air, setting the stage for the Great Oxidation Event. Life that once thrived in dark, anoxic waters faces new challenges and new opportunities. Extinction and adaptation continue, shaping ecosystems in ways that will ripple through billions of years of evolution. This chapter in Earth's history is a study in paradox. The same oxygen that killed some microbes also enabled the evolution of new forms of life. The rusting seas, once inhospitable and corrosive, became fertile grounds for innovation, a crucible where the rules of life were rewritten. The chemical revolutions underway in the oceans were not violent. They were patient, persistent, and relentless, a quiet reshaping of the planet that would resonate for eons to come. And as the BIFs record layer upon layer of these processes, they whisper a warning as well as a marvel. Change on a planetary scale can be subtle, invisible, yet inevitable. The rise of oxygen, wrought by billions of tiny organisms, transformed chemistry, climate, and life itself. The Earth was no longer a static, iron-rich ocean world. It was becoming a dynamic system, capable of supporting complex, diverse ecosystems. So, as we peer into the rust-streaked waters of the Proterozoic, we are witnessing more than a geological phenomenon. We are witnessing a planetary experiment, a chemical drama in which life and environment are inseparably linked. The oceans are a stage, and the actors, microscopic yet powerful, are performing the first acts of a story that will continue to unfold over billions of years. And from this moment of transformation, one question looms. With oxygen slowly escaping into the atmosphere, how would the Earth's climate, chemistry, and life respond? What new worlds would emerge from this chemical revolution? The answers lie ahead, as the stage is set for the next dramatic chapter in Earth's history. The slow dawn of an oxygen-rich sky, a prelude to the Great Oxidation Event, and the rise of life as we would come to recognize it. The Great Oxygenation Event. Billions of years ago, Earth was a planet in quiet turmoil. Invisible to the eye, a drama of life and chemistry was unfolding, a story that would redefine the very air we breathe. For countless millennia, the oceans had been laboratories of change. Cyanobacteria quietly splitting water molecules, banded iron formations, recording each chemical revolution. But eventually, the oxygen they produced could no longer remain hidden. Slowly, inexorably, oxygen began to seep from the oceans into the atmosphere, and the stage was set for one of the most profound transformations in Earth's history, the Great Oxidation Event. Imagine the world before this moment, skies tinted with hazy clouds, oceans deep and rust-streaked, life thriving mostly in the shadows, hidden from oxygen's corrosive grasp. Then, as if a valve had been opened, oxygen began to accumulate in the air, a gas that would both destroy and create. Anaerobic microbes, the ancient masters of an oxygen-free world, faced a lethal new threat. Billions of tiny lives, perfectly adapted to the dark iron-rich seas, found themselves suffocating, unable to cope with the very molecule that cyanobacteria had been producing for eons. The first wave of extinction swept through these primordial ecosystems, silent, invisible, but monumental in scope. Yet oxygen was not merely a harbinger of death. It was also a catalyst for innovation. The atmosphere began to change in ways that would reshape Earth itself. With the rise of oxygen, the composition of the air was no longer dominated by methane alone. Methane, a potent greenhouse gas, began to break down under the influence of oxygen, triggering a cascade of consequences. The planet, once swaddled in warmth, began to cool. Ice may have crept toward the equator, and in these chilling conditions, life faced new challenges and new opportunities. Earth wobbly, as, in essence, adjusting to its own invention. This was a planet in feedback, a system responding to the activities of its smallest inhabitants. Chemical reactions in the atmosphere, driven by the accumulation of oxygen, influenced climate, which in turn influenced life, which then altered chemistry further. It was a delicate, intricate dance, a planetary conversation written in molecules and light, stretching over millions of years. Visualize the sky. Oxygen molecules accumulating, invisible but potent, forming the first protective ozone layer. This shield, faint at first, began to block harmful ultraviolet radiation from the sun, creating pockets of safety for life to venture into previously hostile environments. The Earth's surface, once battered by radiation and corrosive gases, was becoming habitable in ways never seen before. Oxygen, though deadly in one context, was also a guardian, an architect of new possibilities. Meanwhile, the oceans continued their slow transformation. Iron deposits dwindled as oxygen reacted with remaining dissolved minerals, the rusting layers fading as the chemical composition of the water shifted. Anaerobic microbes retreated to oxygen-free niches, while those capable of tolerating or even exploiting oxygen began to diversify. The seas, once monochrome in chemistry, became stratified arenas of contrasting environments, oxidized surface waters, teeming with new metabolic possibilities, deeper anoxic layers, sheltering ancient lineages. This was not a sudden upheaval, but a drawn-out revolution. The oxygenation of the atmosphere unfolded over millions of years, a slow but relentless process that left its mark in rock, ice, and sediment. Geological evidence shows us that oxygen levels fluctuated, rising and falling, yet the overall trend was unmistakable. Earth was being transformed from an anoxic alien world into one capable of sustaining complex life. Consider the paradox. Life itself had created the very conditions that could threaten it. Cyanobacteria, tiny as they were, had engineered a world inhospitable to the microbes that had come before. Yet this same oxygen would eventually pave the way for the rise of complex organisms, for the emergence of eukaryotes, and ultimately, for the entire tree of life that we inhabit today. The story of the Great Oxygenation Event is one of both destruction and creation, a reminder that planetary scale change often arises from the smallest of actors. As the climate shifted, feedback loops intensified. Cooling oceans affected circulation, altering nutrient distribution, and the availability of chemical compounds vital for life. Oxygen levels continued to rise, reinforcing chemical transformations in the atmosphere. The Earth was no longer passive. It was responsive, a system where biology and geochemistry were inseparable partners. Life had become a force of planetary engineering, reshaping the air, water, and surface itself. Imagine standing on this ancient shore. The ocean stretches before you, once rust-streaked, but now clearer, the surface glinting under a pale sun. Above, the sky is gradually changing, oxygen mixing into the air, the first protective ozone shimmering faintly. The world is in transition, chaotic yet purposeful, hostile yet pregnant with possibility. Each bubble rising from the ocean, each molecule reacting in the atmosphere, tells a story of slow, patient transformation on a scale difficult for human minds to grasp. The Great Oxidation Event was more than a chemical milestone. It was a turning point in Earth's narrative. It marked the moment when life asserted control over the planet in a visible, irreversible way. From the microscopic toil of cyanobacteria to the sweeping transformation of atmosphere and climate, this period reminds us that Earth is a dynamic, living system capable of self-transformation. What began as a microscopic process in rusting seas became a planetary revolution, leaving an indelible mark in rocks, sediments, and the very air we now breathe. And yet, even with oxygen accumulating, the story was far from complete. Life had more chapters to write, more boundaries to push, and more experiments to perform. The stage was set for the evolution of more complex, energy-demanding organisms. With an oxygenated sky, new forms of metabolism could emerge. The potential for innovation in biology was unprecedented. Earth had changed, but its story was only beginning. So, as we reflect on this monumental era, one question hangs in the balance. How would life respond to its own planet-altering invention? What new ecosystems, new metabolisms, and eventually, what new creatures would arise from a world now shaped by oxygen? The answers await in the next chapter, where we witness the slow emergence of complex cells and the dawn of eukaryotic life, a prelude to the ever-branching tree of life that would eventually include us. The Great Oxygenation Event stands as a testament to the power of the smallest organisms to reshape a planet. It is a story of paradox and possibility, of death and creation intertwined. Earth, once an alien world of dark, iron-rich seas, was now awakening under a sky gradually suffused with oxygen, preparing for the next grand act in the drama of life. And as the oxygenated air settled over the oceans and continents, a new era loomed, a time when life would move beyond the microbial, when cells would grow larger, more intricate, and more ambitious. The story of oxygen was just the beginning. What comes next is a tale of complexity, adaptation, and the first steps toward the world as we know it, a world waiting to be explored in the chapters that follow. Changing skies. Billions of years ago, the sky above Earth was not the vast blue expanse we take for granted today. Instead, it was a murky ochre haze, thick with dust, volcanic gases, and chemical byproducts from oceans teeming with unseen microbial life. Sunlight filtered through this veil, like amber through fog, casting a muted glow across rust-colored seas. Yet even in this dim alien light, Earth was beginning to undergo a subtle transformation, one that would reshape the relationship between life and the heavens above. The Great Oxygenation Event had already begun rewriting the rules of life below, but it was also quietly altering the skies. As oxygen accumulated in the atmosphere, it reacted with ultraviolet radiation in the upper layers, gradually forming the first ozone. This thin, protective layer began to filter the sun's most harmful rays, shielding emerging life from destructive ultraviolet light. The hazy brown-orange canopy of earlier eons was slowly lifting, giving way to a cleaner, clearer sky, one that hinted at the pale blues of a world in transition. Imagine standing on the shallow edge of a Proterozoic sea. Beneath your feet, microbial mats ripple gently in oxygenated water, each layer a miniature ecosystem shaped by chemistry and sunlight. Above, the sky begins to lighten, a veil of ozone shimmering faintly against the sun. For the first time in billions of years, life could venture closer to the surface without being scorched by radiation. The very air itself had become a protective ally, allowing microbes to explore new environments, stretching from shallow pools to the margins of emerging landmasses. This transformation of the sky was far from superficial. The presence of ozone not only shielded life, but also opened new possibilities for evolution. Shallow waters, previously inhospitable due to intense ultraviolet exposure, became potentia, habitats. Microbes that had once been confined to the depths or hidden beneath mats could now colonize the upper layers of the oceans. These pioneering organisms began to experiment with new forms of photosynthesis, growth patterns, and metabolic pathways. Life was stepping cautiously into a broader world, testing the boundaries of possibility in a universe shaped by chemical change. But the implications extended beyond the oceans. Tiny islands, coastal margins, and the first exposed land surfaces became potential havens for microbial colonization. The interplay between water, air, and light created micro-environments that encouraged diversity and complexity. Life was no longer strictly underwater. The edges of the continents were becoming laboratories for experimentation, places where early organisms could probe the limits of survival and adaptation. The visual effect of this sky-changing revolution would have been mesmerizing. The familiar blue hue of scattered sunlight began to emerge as oxygen concentrations increased, interacting with atmospheric particles. The once oppressive amber-brown glow lifted, replaced by pale blues that deepened toward the zenith. Sunlight penetrated the waters more efficiently, energizing photosynthetic microbes and reinforcing the cycle of oxygen production. A feedback loop of light, chemistry, and life was now in full swing, each element amplifying the others in a planetary symphony of change. Yet even as the sky cleared, the Earth remained a world of extremes. Volcanic eruptions still injected particles into the atmosphere. Occasional ice ages chilled the planet, and chemical imbalances continued to ripple through oceans and soils. Life's expansion into this newly illuminated world was cautious, tentative. Each microbial colony, each shallow water mat, represented both a triumph and a gamble. The environment was gradually more forgiving, but survival still demanded ingenuity and resilience. Consider the role of ultraviolet radiation in this transitional period. Before the ozone layer, life was constrained, largely confined to the protective depths of oceans. With the arrival of ozone, UV radiation was no longer a universal killer. Some organisms even began to exploit the energy in sunlight, using it to fuel metabolic reactions. Pigments evolved to absorb or deflect light, giving rise to dazzling microbial colors in shallow pools and coastal zones. Earth's first visual landscapes were no longer monotone. They glimmered with chemical and biological activity, a subtle hint of the beauty to come in later eons. The changing sky also influenced atmospheric circulation and climate. As oxygen levels rose, the balance of greenhouse gases shifted. Methane, once abundant, began to break down more rapidly, influencing temperature and weather patterns. Cooler, more stable climates began to emerge in some regions, creating niches for life to expand. Microbes that could tolerate these new conditions thrived, while others retreated, confined to pockets where chemistry still mirrored older times. The sky and surface were now intimately connected, a reminder that planetary evolution is rarely linear, often punctuated by feedbacks and interdependence. As we peer into this world, it becomes clear that the transformation of the sky was not merely a backdrop for life. It was an active participant in evolution. Oxygen, sunlight, ozone, and water worked together to create conditions that were increasingly hospitable, yet still full of challenge. Life's ability to respond, adapt, and exploit these changes was the prelude to a far more dramatic chapter. The emergence of complex cells, the eukaryotes, whose energetic metabolism would allow them to build larger, more intricate bodies. The clearing of the sky had set the stage for this next leap, a slow, patient escalation of complexity that would echo through billions of years. And yet, the story is subtle, almost imperceptible. To our modern eyes, the difference between a hazy brown sky and a pale blue one might seem minor, but for life at the margins of shallow waters and exposed land, it was monumental. It meant access to light, energy, and new habitats. It meant protection from the sun's deadly ultraviolet rays. It meant that evolution could experiment on a scale never before possible. The sky itself had become a canvas for the artistry of life, a medium through which biology and chemistry could collaborate on a planetary scale. As the Proterozoic world continued to evolve, life began to sense the opportunities above the ocean's depths. Shallow pools warmed under the filtered sunlight, microbial mats thickened and diversified, and the edges of continents became exploratory zones. Oxygen, once deadly to many, was now a gateway to innovation. The sky, transforming from a hazy veil to a protective and energizing dome, had rewritten the rules of the game. What had begun as a slow chemical accumulation was now a structural feature of the planet, shaping the trajectory of evolution in ways both subtle and profound. So, as we gaze upward at the clearing skies of this ancient Earth, a question arises. How will life respond to this newfound freedom, this opportunity to expand, diversify, and innovate? The answer will unfold in the next chapter, where the first eukaryotic cells emerge, organisms capable of harnessing energy with unprecedented efficiency, exploring the shallow waters, and eventually laying the foundation for all complex life that follows. The transformation of the sky was more than a visual shift. It was a fundamental recalibration of life's environment. By filtering radiation, allowing light to penetrate the oceans, and stabilizing climates, the atmosphere itself had become an architect of possibility. Earth was no longer simply a watery, rust-streaked world. It was a planet with potential, a world increasingly receptive to complexity, experimentation, and the slow march toward the diversity we recognize today. And so, as the hazy orange-brown veil lifts and pale blue stretches across the horizon, we sense the dawn of a new era. Life has been given a new canvas, a broader stage upon which to innovate. The next act is already stirring beneath the surface, in the first complex cells, ready to seize the advantages of an oxygenated, illuminated world. The skies have changed, and with them, the possibilities for life have expanded beyond imagination. The stage is set. The first eukaryotes are on the horizon, and with them, the story of Earth's complexity will take its next great leap. Microbial cities beneath the placid surfaces of shallow Proterozoic seas, a quiet revolution was taking place, one invisible to the naked eye, yet monumental in its consequences. Life, still microbial and single-celled, was beginning to engineer the world around it. Across these ancient waters, colonies of microorganisms were weaving intricate, layered structures known as microbial mats. In some places, these mats built up over millennia into stromatolites, towering column-like edifices of sediment and living tissue. These were Earth's first cities, not of stone and steel, but of cells and chemicals, alive and growing, reshaping their environment in ways that would echo for billions of years. Imagine a shallow lagoon bathed in filtered sunlight. At the top of these microbial mats, cyanobacteria capture photons, splitting water molecules and producing oxygen as a byproduct. Below, other microbes recycle nutrients, feeding on organic matter and chemical gradients created by their neighbors. Each layer is specialized, a vertical economy of life, where energy flows from light at the surface, down through chemical reactions in the depths. This is a world of order emerging from chaos, where life is not merely surviving, but engineering its surroundings. Stromatolites are both biological and geological wonders. Their layered columns, sometimes meters tall, grow slowly over centuries. Each layer records the rhythms of microbial life, the seasonal shifts, the chemical pulses of the surrounding waters. To modernize, they are fossils, static stone. Yet at the time of their formation, they were dynamic, living skyscrapers, teeming with microbial activity. These structures were not just habitats, they were ecosystems, shaping and regulating the chemical composition of their environment. Within these microbial cities, light and chemistry created zones of incredible diversity. No bacteria at the top harnessed sunlight for energy, producing oxygen and organic carbon. Beneath them, layers of sulfate-reducing bacteria converted chemical compounds, releasing hydrogen sulfide and recycling nutrients. Methanogens processed byproducts, while iron-oxidizing microbes left subtle streaks of rust-like minerals. Each stratum was both a home and a chemical reactor, creating gradients that allowed life to flourish in multiple dimensions. The microbial mat was a miniature planet in miniature, a model of how life can alter its surroundings. Stromatolites also shaped their environment on a grand scale. As they grew and accumulated sediment, they influenced water flow, trapping particles and altering the chemistry of the surrounding seas. Their very existence modified habitats for other microbes, creating niches that did not exist before. In essence, these microbial architects were transforming the Proterozoic seascape, creating structures that were both shelter and chemical workshop, altering the evolutionary possibilities for generations of life. It is humbling to realize that these microscopic architects were laying the groundwork for planetary change. The oxygen being released at the surface of microbial mats contributed to the slow accumulation of oxygen in the oceans and, eventually, the atmosphere. The chemical zoning within stromatolites created pockets of reduced and oxidized compounds, influencing local and global geochemistry. These tiny organisms were, unknowingly, shaping Earth's climate, chemistry, and habitability. Life at the microbial level was already a planetary engineer. Visualize the textures and colors of these microbial cities. The top layers glimmer with green-blue hues from photosynthetic pigments, streaked with golds and browns where minerals accumulate. Below, dark sulfur-rich layers shimmer faintly with iridescence, reflecting chemical gradients. Water slowly moves through the mats, carrying nutrients, oxygen, and waste products in a continuous circulation that mirrors the flow of life itself. It is a slow-motion ballet, invisible to the casual observer, yet fundamental to the evolution of the biosphere. Stromatolites were not uniform. Each colony adapted to its specific environment: sunlight exposure, water depth, chemical composition, sediment influx. Some grew in flat mats along tidal flats. Others towered in shallow lagoons, responding to currents and waves. Each colony represents an experiment in life's ability to manipulate matter, to build structures that are both functional and enduring. These are the first examples of ecological engineering, a concept we now see on vast scales, in forests, coral reefs, and human cities. Yet here, it began on a microscopic level. The interplay between biology and geology in these microbial cities demonstrates a profound truth. Life does not merely adapt to its environment. It actively shapes it. The accumulation of oxygen at the surface of microbial mats contributed to banded iron formations, while chemical gradients influenced sediment deposition. These living structures became geological markers, leaving a fossil record that tells us not just about life, but about the invisible chemical rhythms of early Earth. In other words, by studying these microbial cities, we are reading the planet's diary, written in layers of stone and cell. Beyond their chemical and geological impact, stromatolites provide insight into evolutionary innovation. The compartmentalization within microbial mats may have facilitated cooperation between cells, laying the groundwork for more complex forms of life. Communities that could maintain chemical gradients, share metabolites, and protect sensitive organisms from environmental stress were more likely to thrive. These mats were early experiments in collaboration, resource management, and environmental manipulation, a prelude to multicellularity and the intricate ecosystems that would follow. Yet, the story of microbial cities is also a story of resilience. Life persisted through fluctuating conditions, chemical extremes, and changing ocean compositions. As oxygen began to rise in the atmosphere, many anaerobic microbes faced extinction, but others adapted, finding refuge within the protective layers of mats or in specialized chemical niches. The layered architecture of stromatolites allowed life to endure, diversify, and continue shaping its environment even amidst planetary upheaval. These structures are living lessons in adaptation and persistence. As we contemplate these ancient microbial cities, a question arises. Could life anywhere else in the universe follow a similar path? Could extraterrestrial oceans harbor microbial mats, slowly engineering their environment, leaving chemical fingerprints of life for billions of years? The stromatolites of Earth suggest that even the smallest organisms can have planetary consequences. They teach us that life is not passive. It is an active participant in the shaping of worlds. As the eons passed, microbial mats continued their work. Oxygen released at their surfaces seeped into the oceans, feeding the slow accumulation that would transform the atmosphere. Their chemical zoning influenced mineral deposition, sediment dynamics, and nutrient cycling. Life, once confined to invisible cells drifting in oceans, had learned to construct, organize, and engineer, a lesson that would echo in all future ecosystems. And yet, even as these microbial cities thrived, the stage was being set for the next revolution: the emergence of complex cells, the eukaryotes. These first architects of the microbial world had created the chemical and structural scaffolding necessary for larger, more sophisticated life forms. The story of life's ascent is a story of preparation, of layers and gradients, of ecosystems built brick by microscopic brick. The lesson is profound. From the smallest cells, Earth learned to build, to organize, and to sustain life on a planetary scale. Stromatolites are not just fossils. They are monuments to microbial ingenuity, testaments to the invisible work of life shaping its world. And as the first eukaryotes emerge in the next chapter, they will inherit the cities, the oxygen, and the chemical landscapes crafted by these tireless microbial engineers. The Proterozoic seas are alive with structures that are part biology, part geology, and entirely a testament to life's capacity to transform the planet. The microbial mats and stromatolites of this era show us that complexity does not always begin with large bodies or brains. It begins with organization, persistence, and the subtle, patient work of millions of tiny cells acting together. And so, as we leave these microbial cities behind, we glimpse the future of life on Earth, a future shaped not just by chance, but by the deliberate, quiet engineering of life itself. The first eukaryotic cells are waiting in the wings, ready to take the foundations laid by microbial cities and build a world of even greater complexity, chemical battles, and biological adaptation. Billions of years ago, Earth faced a paradox, a life-giving gas that was also a silent executioner. Oxygen, the very molecule we now take for granted, had begun to accumulate in the oceans and atmosphere. For some microbes, it was a deadly poison, a chemical threat that disrupted delicate biochemical pathways honed over eons. For others, it represented opportunity, a new energy source, a means to grow faster, to reach farther, to dominate the world. This dual nature of oxygen set the stage for a period of both extinction and innovation, a crucible in which the next chapter of life would be forged. Imagine the oceans at this time, vast, stratified, some layers still anoxic and iron-rich, others beginning to glow faintly with oxygen's presence. Microbial communities, previously adapted to life without oxygen, were now confronted with an invisible chemical onslaught. Enzymes that had functioned flawlessly for billions of years were suddenly poisoned, metabolic pathways disrupted, and entire populations faced collapse. Extinction was widespread, yet it was selective. In the crucible of chemical stress, life began to innovate. Some microbes evolved defenses, specialized enzymes capable of neutralizing oxygen's toxicity. Others found refuge in oxygen-free pockets within sediments, mats, and microbial colonies. Yet, a revolutionary adaptation was quietly emerging, the harnessing of oxygen as a potent source of energy. The first aerobic organisms discovered a biochemical shortcut that transformed this reactive gas into a tool rather than a threat. By coupling oxygen to the breakdown of organic molecules, they extracted far more energy than anaerobic pathways allowed. This was not merely survival. It was a leap in metabolic sophistication. Aerobic respiration changed the rules of life. Microbes that could use oxygen to drive their metabolism grew faster, reproduced more efficiently, and colonized new ecological niches. The energy surplus allowed for larger cell sizes, more complex structures, and the development of regulatory systems. Life had learned to turn poison into power. And in doing so, these pioneering cells laid the groundwork for eukaryotic life, the line of cells that would one day give rise to all complex organisms, including plants, fungi, and animals. Yet this was not a gentle transition. The rise of oxygen precipitated a global crisis. Anaerobic microbes, unable to cope with the increasing concentration of oxygen, faced massive die-offs. These chemical battles reshaped the biosphere, resetting ecosystems and opening niches for organisms that could thrive in oxygenated conditions. It was a period of paradoxical creativity born from destruction, extinction fueling innovation, mortality driving evolution. The oceans themselves became a mosaic of chemical zones. In the depths, oxygen was scarce. Iron and sulfur reactions dominated, and anaerobic life persisted in hidden refuges. Near the surface, oxygen levels rose, enabling aerobic metabolisms to flourish. Microbes adapted to these stratified conditions, evolving specialized lifestyles that exploited gradients in oxygen, light, and nutrients. The Proterozoic seas were now a dynamic theater of chemical warfare and biological ingenuity, where survival demanded both resistance and opportunism. Consider the role of oxygen in energy production. A single molecule of glucose, processed anaerobically, yields only a small fraction of the energy it can produce in the presence of oxygen. With aerobic respiration, cells could generate 10 times the ATP, fueling more elaborate biochemical machinery. This energetic revolution permitted structural complexity and cellular differentiation, critical prerequisites for the eventual evolution of eukaryotic cells. The tiny architects of the microbial mats had built the foundations, but oxygen provided the energy currency to construct the next level of life. Chemical adaptation was not limited to energy production. Microbes developed protective enzymes such as superoxide dismutase and catalase, molecules capable of neutralizing the destructive reactive oxygen species that threatened cellular integrity. Life learned to engineer biochemistry itself, to defend against a reactive environment while exploiting it for growth. These innovations illustrate a profound principle. Stress drives adaptation, and adaptation drives the evolution of complexity. The story of microbial adaptation to oxygen is also a story of planetary feedback. As aerobic microbes multiplied, they accelerated the consumption of reduced compounds in the oceans, further altering chemical gradients. Oxygenation of surface waters increased, feeding back into atmospheric composition. Life was no longer merely reacting to Earth's chemistry. It was actively reshaping it, preparing the planet for forms of life that required not just energy, but stability and chemical diversity. The stage was being set for eukaryotic cells, capable of compartmentalization, internal energy management, and eventually multicellularity. Imagine the silent struggle in these ancient seas, microbial populations clinging to survival, some thriving in newfound oxygen-rich zones, others hiding or dying off. Chemical gradients formed invisible front lines, boundaries where metabolic strategies collided. Life was not a passive passenger on Earth. It was a chemical engineer, a strategist, and an innovator, learning to turn adversity into opportunity. This period, though invisible in the fossil record, was crucial, a hidden drama that determined which forms of life would persist and which would vanish. It is here, in these chemical battles, that the first hints of eukaryotic evolution appear. Organisms capable of exploiting oxygen had access to unprecedented energy reserves. Some may have engulfed other cells in symbiotic relationships, eventually giving rise to mitochondria, the powerhouses of eukaryotic cells. These early partnerships, driven by chemical opportunity, would revolutionize life, allowing for greater cellular complexity, specialization, and resilience. Oxygen, once a deadly poison, became the architect of biological innovation. And yet, life did not simply survive. It transformed. Microbes engineered not just chemical solutions, but ecological structures. Microbial mats and stromatolites continued to thrive, now layered with oxygen-producing cyanobacteria at the surface and oxygen-tolerant heterotrophs below. These layered ecosystems were living laboratories, incubators for adaptation and innovation. In the shadows of these mats, eukaryotic precursors would eventually find niches to exploit, pre-adapted to a world where oxygen was both threat and resource. The rise of oxygen also reshaped global climate. Methane, a potent greenhouse gas, abundant in the anoxic Proterozoic atmosphere, was broken down in the presence of oxygen, cooling the planet and triggering glaciations. Life and chemistry were intertwined, each influencing the other, each feeding back into global processes. Earth was a laboratory of chemical experimentation, and the participants were the tiniest of organisms. As we peer into this ancient, oxygen-struggling world, we see more than extinction and adaptation. We see preparation. Microbes were not only surviving, they were setting the chemical and energetic foundations for a revolution in cellular architecture. They were readying the oceans for eukaryotic cells, the next protagonists in the story of life. Complexity was no longer a distant possibility. It was emerging, fueled by chemical opportunity and sculpted by the invisible hand of adaptation. In this period of chemical conflict, life learned resilience, ingenuity, and cooperation. Microbial strategies to survive oxygen stress, to harness its energy, and to exploit chemical gradients demonstrate that evolution is not random chance alone. It is the consequence of persistent innovation under pressure. Oxygen shaped life, and life, in turn, reshaped oxygen. This interplay of chemistry and biology laid the foundations for a world in which large, complex cells could emerge, thrive, and eventually populate land and sea in unimaginable diversity. And so, as the Proterozoic seas simmered with chemical tension, the first eukaryotic cells began to take shape. The stage was ready. Oxygen, once a deadly poison, was now a fuel for complexity. The microbial architects of old had built the infrastructure. The chemical battles had set the rules. The next act, the rise of complex cells, would transform the story of life forever, carrying Earth from invisible microbial cities to the dawn of organisms capable of shaping their own destinies. The chemical battles of this era remind us that survival is not always about strength. Sometimes it is about adaptation, ingenuity, and the ability to turn threat into opportunity. Life had learned these lessons well, and as the eukaryotes emerged from the shadows of microbial mats and oxygen gradients, Earth was ready for a new chapter, a chapter that would culminate in complexity, diversity, and the astonishing tapestry of life we see today. Climate, ice, and planetary feedback. Billions of years ago, Earth began to reveal another dimension of its power. Climate, not as a background to life, but as a dynamic force shaped by the tiniest of organisms. The rise of oxygen in the oceans and atmosphere had already rewritten the rules of biology, but it also set in motion a chain of planetary events that would cool the planet, trigger massive glaciations, and challenge life in unprecedented ways. To understand the first ice ages, we must look not only at chemistry, but at feedback loops between life and the very air we breathe. Consider methane, a potent greenhouse gas that had long wrapped the young planet in warmth. In the anoxic Proterozoic atmosphere, methane concentrations were high, keeping Earth from freezing even as the sun shone more faintly than it does today. But oxygen, the very molecule that had begun to empower life, also disrupted methane's stability. As cyanobacteria and aerobic microbes multiplied, oxygen broke down methane, thinning the planet's thermal blanket. Suddenly, Earth was cooling. A delicate chemical balance had shifted, and the first planetary ice ages were on the horizon. Imagine this cooling world, vast oceans beginning to chill, polar regions advancing toward icy extremes, and glaciers creeping into continental interiors. These were not mere seasonal snows or modest frost lines. They were global-scale transformations, with ice sheets potentially covering the equator itself. Some scientists call this period Snowball Earth, and it is a reminder of how closely life and climate are intertwined. Oxygen, once a silent threat and then a source of energy, had indirectly plunged the planet into cold, testing life at every turn. Yet ice was not the only story. Carbon dioxide, the other great regulator of climate, was also under the influence of biology. Photosynthetic microbes drew down carbon dioxide from the atmosphere, locking it into rocks and sediments, while chemical reactions with silicate minerals further sequestered this greenhouse gas. With less carbon dioxide and declining methane, temperatures fell even more. Feedback loops, complex self-reinforcing cycles, began to dominate. Cooling oceans, shifting atmospheric composition, and the interplay of greenhouse gases created a dynamic climate system unlike anything Earth had experienced before. For life, these shifts were both perilous and transformative. Microbes adapted, finding refuge in equatorial oceans, deep water pockets, and beneath ice layers. The harsh climate imposed selective pressures that drove evolution forward, favoring metabolic flexibility and resilience. Ice did not merely threaten survival. It reshaped ecosystems, reorganized microbial communities, and set the stage for innovations that would later support more complex life forms. In the struggle against the cold, life learned endurance, patience, and adaptation. Planetary feedbacks during this era reveal a profound truth. Earth is not a passive stage for life. Life actively influences the climate, which in turn shapes life's possibilities. Oxygenation reduced methane, cooling the planet. Cooling led to ice formation, which reflected sunlight and further dropped temperatures, a self-amplifying loop. Even the arrangement of continents played a role, as landmasses near the poles enhanced glaciation, altering ocean currents and weather patterns. Every element was interconnected: atmosphere, oceans, life, and geology, acting in concert to create a planet of constant change. Visualize the Proterozoic Earth from above. A patchwork of ice, dark oceans, and scattered continents. Methane haze fading, oxygenating skies above, and microbial mats clinging to survival in pockets of liquid water. Life was facing extremes it had never known, yet it endured. These ice ages were not mere catastrophes. They were selective forces, sculpting organisms capable of complexity, endurance, and ingenuity. In many ways, they were a proving ground for life itself. The cooling world also affected nutrient cycles. Glaciation enhanced weathering of rocks, releasing essential minerals into the oceans. Iron, phosphorus, and other nutrients flowed into microbial habitats, stimulating growth where conditions allowed. Even as ice threatened survival, it supplied the chemical ingredients for renewal. Life and climate were dancing in a feedback loop, sometimes destructive, sometimes generative. Evolution was no longer random. It was tightly coupled to the planet's chemistry and temperature. One of the most fascinating consequences of these climate shifts was the stage they set for multicellular life. Single-celled organisms had dominated for billions of years, mastering oxygen, chemical gradients, and energy extraction. Yet, the stresses imposed by extreme climate may have nudged cells toward cooperation. In frigid, nutrient-variable environments, multicellularity offered advantages: resource sharing, division of labor, and protection against environmental extremes. Ice ages did not simply shape survival. They created the conditions that made complexity advantageous. The first stirrings of multicellular life were thus inseparable from the icy trials of the Proterozoic. Methane breakdown, oxygenation, glaciation. These were all interconnected threads in a global narrative. Planetary feedbacks amplified subtle changes, turning microscopic biological processes into global scales. The consequence. The first ice ages remind us that life does not merely adapt to the environment. It reshapes it, and the environment, in turn, reshapes life. Each cycle of cooling and warming, each shift in greenhouse gas concentrations, was a lesson for the biosphere, preparing organisms for the challenges and opportunities of the future. Even in the depths of these ice ages, life was not static. Microbial mats persisted, oxygen levels continued to fluctuate, and chemical cycles carried on beneath the ice. These resilient communities maintained the engines of evolution, providing energy, recycling nutrients, and experimenting with new metabolic pathways. The stage was being set for a revolution in cellular complexity. Eukaryotic cells, benefiting from energy-intensive aerobic metabolism and ecological opportunities created by nutrient pulses, were poised to expand and diversify. Ice and chemistry together had primed the planet for life's next leap. And yet, the story of Proterozoic ice is not one of despair. It is a story of feedback, resilience, and transformation. Life endured the cold, and in doing so, prepared for its own ascent. The interplay of oxygen, methane, carbon dioxide, and temperature exemplifies a fundamental principle of Earth. Its systems are deeply interconnected, and even the smallest organisms can wield planetary influence. These ice ages were as much about shaping life as they were about shaping the planet itself. As the glaciers receded and oceans slowly warmed, the chemical and ecological stage was set. Oxygen remained a pervasive force. Life had adapted to extremes, and eukaryotic cells had the energy and resilience to expand into new niches. The Proterozoic ice ages were not the end. They were a prelude to the rise of multicellular life, to the explosion of complexity, and to the emergence of ecosystems that could alter continents and climates. The planet was ready for a new act in the grand story of life. And so, as we leave behind the frozen oceans and turbulent feedback loops of the Proterozoic, we glimpse the next chapter, a world where complex cells, collaborating and competing, would begin to create life forms that could shape not only oceans and skies, but the very surface of the planet. The icy crucible had tested life's limits, but it had also taught resilience, cooperation, and innovation, lessons that would echo into the dawn of multicellular life. From the chilling depths of the first planetary ice ages, Earth was preparing for its next transformation. A transformation that would bring color, motion, and diversity to the microbial cities that had persisted for billions of years. The stage was set, the chemical and climatic foundations were laid, and life was poised to take its next leap toward complexity, collaboration, and the astonishing emergence of multicellular ecosystems. The frozen crucible of the Proterozoic whispers an enduring lesson. Earth is a planet of feedback, of cycles, of invisible threads linking chemistry, life, and climate. And within these threads, the seeds of the next great evolutionary story were quietly germinating. Legacy of the Proterozoic. As we step back from the icy crucibles, the oxygenated seas, and the chemical revolutions of the Proterozoic, a quiet truth emerges. The Proterozoic was not dramatic in the way we often imagine life. No towering beasts, no swarming forests, no dazzling skies filled with birds. It was a time of subtlety, of invisible forces, and of countless tiny actors whose influence would ripple through billions of years to shape the world we inhabit today. To understand the legacy of this era, we must look closely at the microscopic architects of Earth. From the first cyanobacteria that captured sunlight to the microbial mats, constructing towering stromatolites, life was experimenting with possibilities on a planetary scale. Tiny cells, invisible to the naked eye, were producing oxygen, transforming oceans, and even influencing the climate. The accumulation of oxygen in the atmosphere, once a deadly poison for the anaerobic majority, became the foundation for aerobic metabolism, enabling organisms to extract far more energy from their surroundings. With more energy came the potential for complexity, for cooperation, for cellular experimentation that would eventually lead to the first eukaryotes. Stromatolites, those layered microbial cities, were more than biological curiosities. They were the earliest evidence of life shaping its environment. Photosynthetic communities at the surface captured sunlight, while layers beneath recycled nutrients, creating chemical gradients that influenced the surrounding ocean. These structures were literal monuments to cooperation and resilience, standing as silent witnesses to the first billion years of Earth's evolutionary experiment. Look closely at their fossilized remains, and you are looking at the first whispers of planetary engineering. Chemical evolution during the Proterozoic was no less astonishing. Oxygen was accumulating, methane was declining, and carbon dioxide was sequestered in sediments and minerals. Feedback loops between life, atmosphere, and climate were writing a story of balance and tension. The planet had learned, in a sense, to self-regulate, with microbial life as its primary agent. Without these processes, Earth might have remained a warm, stagnant, uninhabitable world, or it could have frozen entirely under the weight of unchecked greenhouse gas depletion. In the intricate dance of chemistry and biology, life became both choreographer and performer. Yet the Proterozoic was not only about survival, it was a time of preparation. The first eukaryotic cells, more complex than their bacterial predecessors, began to emerge during this era. By engulfing other cells, evolving internal compartments, and experimenting with energy-intensive aerobic metabolism, these pioneers laid the groundwork for multicellularity. Every chemical shift, every ice age, and every oxygen spike acted as a selective pressure, nudging evolution forward. The planet was conducting a slow, patient rehearsal for the grand show of complex life that would follow. The oceans, once murky and toxic, were gradually transformed into more habitable realms. Oxygenated waters allowed new metabolisms to flourish, while stratification and nutrient cycling created niches where diversity could expand. Microbial ecosystems were no longer merely surviving. They were shaping the very conditions under which life could evolve further. The subtlety of these changes is staggering. A world ruled by tiny, unseen organisms was methodically sculpting a stage for everything that would come next. Reflect on the planetary feedbacks that defined the Proterozoic. Glaciations, oxygenation, chemical cycling. They were all connected, a network of cause and effect spanning oceans, atmosphere, and land. Life was not passive. It was an active participant in planetary change. The consequences of these interactions are still evident today. Banded iron formations, oxygen-rich sediments, and the very composition of our atmosphere are enduring records of this invisible work. Every breath we take, every molecule of oxygen in our lungs, is a gift from these ancient microbial engineers. And yet, for all its profound influence, the Proterozoic was a world of patient waiting. Evolution's pace was slow, measured in millions of years, and change was often imperceptible in the moment. But beneath the surface of oceans, within the layers of stromatolites, and in the chemical makeup of the atmosphere, a quiet revolution was underway. Life was learning, adapting, and accumulating the energy, complexity, and resilience necessary for a future that would one day burst into visible diversity. The legacy of the Proterozoic is, in many ways, a lesson in subtlety. It teaches us that transformation does not always come in dramatic leaps, that the most profound changes often occur invisibly, and that the smallest actors can shape the destiny of an entire planet. Tiny cells, unseen and silent, set the stage for everything we know today. Without them, there would be no eukaryotes, no multicellular life, no animals, no plants, no humans, to ponder the story of Earth. As we stand at the threshold of what comes next, it is worth pausing to consider the magnitude of this preparation. The Proterozoic was Earth's slow adolescence, a period of maturation where the atmosphere, oceans, and life itself were learning to coexist in a delicate balance. It was a time when Earth discovered the potential of oxygen, mastered feedback loops, and experimented with complexity on a microscopic scale. It was a time when life, though largely invisible, began writing the blueprint for the future. And now, with the Proterozoic drawing to a close, the stage is set for the most visible revolution in the history of life. The chemical and biological foundations are ready. Multicellular organisms are emerging from the shadows of microscopic life, poised to diversify, innovate, and colonize new environments. Ecosystems that once consisted solely of microbial mats are giving way to networks of interacting organisms, each influencing its surroundings in ways more complex than ever before. The next act, the Cambrian explosion, awaits, a period when life would burst into visibility, revealing shapes, colors, and behaviors that would forever transform our perception of the living world. The Proterozoic may have been quiet, slow, and hidden, but it was the crucible in which the possibilities of complex life were forged. Without billions of years of microscopic innovation, the spectacular diversity of animals, plants, and eventually humans could never have occurred. So as we leave behind the slow, patient world of the Proterozoic, we carry with us a profound understanding. Earth's history is not merely a story of dramatic moments and visible creatures. It is a tale of persistence, of invisible labor, of subtle forces shaping the possibilities of life. Tiny cells, unseen and silent, sculpted oceans, altered atmospheres, and rewrote the rules of evolution. They taught resilience, created opportunity, and prepared the world for an explosion of complexity that would captivate the imagination of the universe. The legacy of the Proterozoic lingers all around us, in the air we breathe, the rocks beneath our feet, and the living systems that sustain the planet. It reminds us that even the smallest actors, when persistent and creative, can change the course of history. And as the Proterozoic fades into memory, the Cambrian dawn rises, a period of color, motion, and visible life that will forever redefine Earth's surface and ecosystems. From invisible architects to visible innovators, life was poised to step into the light. And for the first time in billions of years, the stage was ready for organisms large enough to see, complex enough to interact, and diverse enough to shape the very planet in ways that would echo across deep time. The Proterozoic may have been silent, but its story resonates through every breath, every organism, and every ecosystem today. The Nani Dexti chapter is about to begin. The Cambrian explosion, a world where life will no longer hide in the shadows, but will reveal itself in all its dazzling complexity. The tiny cells of the Proterozoic set the foundation, but the stage is now prepared for an unprecedented display, the dawn of visible life.